Welcome, everyone. I'm thrilled to be here today to welcome each and every one of you to this first preliminary webinar for our upcoming workshop, Trust Your Gut, Establishing Confidence in Gastrointestinal Models. It is scheduled for October 11th and 12th. Just some background, the human gut is often referred to as the second brain. It is a very complex ecosystem that plays a pivotal role in our overall well-being. Orally administered substances have the potential to be first metabolized in the GI tract before they are being absorbed into the system. And understanding gut absorption will provide insights into systemic exposures and potential systemic toxicity. Also, new approach methods provide unique opportunities to screen for potential GI tract toxicity, adverse effects on the microbiome, or allergenicity resulting from orally administered substance. NAMs can also offer more human-relevant predictions of potential localized and systemic toxicities. And I would like to acknowledge the organizing committee for their dedication and tireless efforts in making the workshop and webinars possible. Over the next few weeks, we will offer, including today, 3 webinars to lay the foundation for our in-person workshop, where we will delve deeper into the state of the science and context of use of various gut models. Scientists and regulators will come together to discuss topics related to building confidence and explore the strengths and limitations of these model systems. So please remember to register for the second webinar on September 20th and the third webinar on October 6th. And for the workshop on October 11th and 12th, you can register to participate in person or virtually. In today's webinar, our primary focus will be on regulatory perspectives, featuring distinguished speakers from both the US EPA and the FDA. Just a quick note on housekeeping. You have the option to change your display name in the participants window, and you can find a toolbar at the bottom of your screen if you want to ask questions. And if time permits, speakers will take clarifying questions after the talk, and you can use the raise hand option to speak, or you can type your question in the Q&A tab. This webinar is being recorded, and the recordings will be made available at the NIEHS website. So thank you very much for being here today, and let's get started. Okay, let me introduce our first speaker, Dr. Maureen Nguyen, who is the Principal Deputy Assistant Administrator for the Office of Research and Development at the US EPA. She provides direction on overall program goals, objectives, policies, strategies, technical and scientific approaches, and program plans. She also chairs the agency's Science, Technology, and Policy Council and oversees the agency's Scientific Integrity Program. Her talk today is on development of new approach methods for decision-making. Maureen?
There we go. Sorry about that. I have a new setup at home and I'm not used to Zoom. Thank you, Cecilia. I appreciate it. So this is my new approach method, is trying to use Zoom on my new setup. So that is the focus of my conversation today. Thank you very much for the invitation to be part of this webinar series and actually to be kicking it off. I really appreciate being able to not only be here today and hopefully listen into some of the other webinars, and then hopefully some of the workshop, though that'll be a little bit of tough time in terms of 2 full days. But I'm very interested in learning more about the direction things are going related to gastrointestinal models. So as Cecilia said, my presentation today is development of new approach methods for decision-making. And also as she said, at the US EPA. This will be a very high-level overview of some of the general considerations for NAM development. And, I think the overall thrust of these webinars is not only the background, but also to talk about what directions we might need to go into to build scientific confidence in our new approach methods, specifically in this case to GI models, but even beyond that. And I think for the EPA context and the regulatory lens, it will not be as specific to the gut or the GI models in my presentation today, but I think a lot of what we've been thinking at the EPA and the direction we need to go in the field for general sort of increasing acceptance of the use of NAMs is transferable to these specific models and topics that will be discussed in future webinars and at the workshop. So looks like everything's moving forward. So I think I've passed that first test. So this is a really old school slide on new approach methods or NAMs. I keep using it, even though I've been using it since I think 2019 or so, because there is no specific formal definition of NAMs in the federal government or at the EPA. So I like to put it into context in terms of what I'm thinking and what we're thinking when we talk about NAMs. The definition I have here is the first bullet is what we had put in one of the documents on the left showing the strategic, it was a TSCA report looking at strategic planning related to the use of NAMs or alternative toxicity methods. And then some of the terminology also came from a 2016 ECHO workshop, which is the other workshop report shown to the left. So generally NAMs are defined as any technology, methodology, approach, or combination that can provide information on chemical hazard and risk assessment to avoid the use of animal testing. The second bullet is sort of where we expanded a bit to commonly define, including in silico approaches, in chemico and in vitro assays, and then making sure we are including information on exposure of chemicals in the context of hazard assessment. I think this is not that dissimilar from other definitions of NAMs, but again, there are some variations depending on the context and the focus of the work that's being done. The challenges listed here, these are challenges we face across the board when we're talking about building confidence in the science that is the foundation of regulatory decision-making. We have multiple decision contexts, not only just at the EPA, but between the EPA and FDA and other federal agencies, particularly the regulatory agencies. We have a lot of different statutes and mandates that we must adhere to. Also, again, building that scientific confidence in the use of NAMs in regulatory decision-making. This is a requirement for all methodologies used to support regulatory decision-making. But our focus now obviously is how do we build that as we're sort of building and developing new approach methods? How do we look at what we're doing in a way that will help increase scientific confidence? And then finally, addressing complex issues. Again, this is not just specific to NAMs, but in the NAMs space, how can we use these new approach methods to address things like cumulative impacts and thinking about mixtures, and how is this new methodology able to help us increase that effort? So I was recently engaged in another activity in the federal government that was asking the question of where and how do we focus our resources for future NAM development? How are federal agencies in particular doing that? And what are the key things to consider? So I co-opted this slide from a presentation I gave there about how EPA is thinking about it, because I thought as I was putting this talk together, this was some of the lens that I had in terms of how are agencies prioritizing and integrating NAMs into scientific aims and or regulatory processes, like bringing this back to the need for scientific confidence. That's the underlying factor in what we're trying to do, what we're trying to develop, how we validate, how we evaluate performance, so that what we're putting our resources and time and energy into is something that's usable in supporting the work that we're doing to protect public health and the environment for EPA. Then we also were asking the question of where are new tools or capabilities particularly positioned to complement and or replace traditional models. So this in some ways could be, starting at this level, the low-hanging fruit, right? These are the things that are available or close to available that could really help build the confidence in the use of NAMs as a field, because these are already in existence and already have some confidence in them, ideally. And then finally, what are the biggest challenges in the development and use of NAMs from a regulatory perspective? And again, as Cecilia said at the beginning, both talks today are going to have that regulatory perspective, but I think a lot of our thinking is applicable across the board, regardless of well, the decisions being made or the work that you're doing. And when we talk, although I'm speaking on behalf of EPA here, the regulatory decisions aren't just at the federal level, they're also state and local government. And then working cross internationally in terms of what information is needed to help support the use of NAMs. So at EPA, I won't assume that you're all familiar with the EPA NAMs work plan, but it's been a big part of my life in the past few years. So the EPA NAMs work plan that was first released in 2019 and then updated in 2021. And this is the way that EPA has approached looking at what we need to do in the new approach methods space. There's 5 key objectives, and I'll speak about each of them a little bit before I move on to talk about what we've done since these plans were developed. The 5 objectives were related to reducing animal testing and research while ensuring that agency decisions remain fully protective of human health and the environment. The 5 objectives, as I said, shown here, they're obviously in order because you always have to have a list, but they are being done in parallel. They are not contingent on any one of the other. So some have moved forward and some are ongoing, but we'll talk about some of the details of that in just a moment. But the first is evaluating regulatory flexibility for accommodating NAMs. And so what does that mean? We don't all have a Lautenberg Amendment for all of our regulations where it says basically, look here, you should use NAMs. So what we wanted to do is have a better understanding of what our regulatory, what our statutes, what our policies, what they say related to, if not specifically alternative tox methods, but specifically about traditional toxicity methods. So is there a requirement in some of those where you need to use X assay or these animal models, et cetera, et cetera? And what would that mean if we were trying to move forward on incorporating and integrating NAMs? For the second objective was developing baselines and metrics for assessing progress. This is very clearly, very specifically linked to reducing animal usage. And so that is something that, of course, we can't see if we've made any progress if we don't know where we're starting. The third is establishing scientific confidence and demonstrating application. And I think that's a lot of why we're here. How do we do that for the various types of methods and models and new approach methods? I'll speak more again in some later slides about our EPA scientific confidence framework that we're working on. And then also one that has just been released for public comment from ICFAM. And then the fourth is developing NAMs that fill critical information gaps. And I think third and fourth, not that the others aren't also equally important, but these are, I think, key to the workshop's goals in terms of how do you decide which NAMs to develop to fill those critical information gaps, and what is the importance of understanding the decision context that they'll be used for. And then finally, engaging and communicating with stakeholders. I often think of this as with many things that we do, it will take a village. This is something that cannot be done in silos, although we all have our own specific sort of lane that we're in terms of the different regulations or the different questions and decisions to be answered. But we can work together and not duplicate efforts and really build on each other's efforts across the federal government, across states, academia, industry, and international support. So there we go. So the status of the NAM work plan deliverables, I won't beat this. I won't spend too much time on these, but just wanted to give you a heads up. We're moving forward on a lot of the actions in the EPA NAM work plan, and these are in the same order that I had just read the 5 objectives. So I would note that for the evaluating regulatory flexibility for accommodating the use of NAMs, we've just completed a draft report on what that might look like, and this is specific to EPA. We weren't looking at regulatory flexibility across the federal government, but just at EPA at this point. And it's currently entering internal review. For the developing baseline and metrics for assessing progress, again, we have a progress and summary metrics on reducing vertebrate animal testing requests and use, and that is released annually starting as of last fiscal year. Looking at establishing scientific confidence in NAMs and demonstrating application to regulatory decisions. The first deliverable was released earlier this year. It was a U.S. National Academies of Sciences, Engineering, and Medicine study that evaluated the variability and relevance of existing mammalian toxicity tests and reviews frameworks for validation and establishing scientific confidence in testing methods. And this was funded by the EPA under the NAMS work plan in order to really help get an external view on some of the questions that we had related to scientific confidence in the use and application of NAMS. Some of the sort of abstracted recommendations from the NAS report included broadening the definition of NAMS to include a range of strategies and approaches, establishing performance of NAMS based on intrinsic performance characteristics and protection of human health rather than variability of animal tests, not using animal tests as the sole determining factor in determining internal and external validity, broadening the considerations for evaluating concordance between, beyond comparisons to animal tests. So there's a little bit of a theme here, I think, in some of the recommendations. And then when evaluating a test method where there is no animal human data, acceptance should take into account the experimental and biological variability and internal and external validity. And then finally, developing and utilizing a framework for hazard identification and deriving toxicity values protective of public health that does not require human epidemiologic or animal toxicity data. So a lot of these that I sort of pulled from the recommendations are tied to, I think what a lot of us in the field have said is that if we're doing the new approach method development and validation, a lot of that is being done on chemicals in our case, and where there is not existing data. And so it's not going to be possible to validate the results to existing animal tests or to human studies. And so building that scientific confidence framework on how we evaluate the performance of these names, particularly with data poor chemicals, is going to be very key. Other deliverables here, as I mentioned, is we're working on a scientific confidence framework to evaluate the quality, reliability, and relevance of NAMs. And I do think I have another. Yes, sorry. There's the cover here to the NAS report, and we'll make sure the link is shared if you do not have it already. For our scientific confidence framework, we presented the key principles shown. Oh, I'm sorry. I don't know if you can see my arrow shown here in the bottom of the screen at the 2022 EPA biennial NAMS workshop. And this is expected, as you can see, the deadline, the due date in Q4 of 2024. We're also working on an initial set of reporting templates, which may be used by EPA and other stakeholders to capture the range of specific NAMs used in agency decisions. And this will be key to talk about, to have a better understanding of what NAMs are being used and how they're being used, and that will help us prioritize future efforts in this space. Oops, sorry, let me go back on this one. So yes, okay. So and also case studies for evaluating application to risk assessment and demonstrating protection of human health and the environment. And these are ongoing. The plan was not to do a large number of case studies towards any certain year, but to do them throughout the process. And so that is, as I said, ongoing. I think case studies are a great way to really see what works and what doesn't, and to build sort of a better machine, honestly, in terms of new approach methods. Finally, the last two objectives, developing NAMs to address scientific challenges and fill important information gaps. Within the EPA, we every 4 years do strategic research action plans, which outline research products, and in this case, have quite a few to develop and apply NAMs. That will be something that, it's also ongoing. And then encouraging development of NAMs through mechanisms such as our Science to Achieve Results grant program and facilitating partnerships with organizations focused on establishing scientific confidence in alternative methods. And then for the last objective, looking at these deliverables, as I said, it'll take a village, and the village does better when it communicates, right? So we need to engage and communicate and educate stakeholders on what we're doing and what NAMs are and what they are not. We started off with setting up a easy to find EPA website to house information about NAM efforts, epa.gov/NAM. We have a series of public webinars, and with some of the deliverables for the work plan, they're in peer-reviewed journals or reports available to the public. And then also we started a NAMS pilot training program and working on completing that this year. And we provide regular scientific exchanges and progress updates through agency-sponsored and partner-organized events. It's not too late to register for our next training on HTTK, which will be November 8th through 9th in 2023. And the link shown here is where you can go to register for that. And you can also look for recordings of previous trainings. So shifting gears a little bit from all sort of the housekeeping of deliverables, but I guess to summarize those lists, EPA is really invested in advancing new approach methodology and the use for our regulatory decision making. And I think that's clear from from the development of the NAMS work plan and also the work that's been done in the past few years. The implementation team has done a great job of moving the ball forward on that. And really keeping people abreast of what is happening in that space and what some of the future needs might be. So we are working, as I mentioned, as one of the deliverables on a NAMS confidence framework. Some of the NAMS work plan implementation team members developed with ICFAM the white paper validation, qualification, and regulatory acceptance of new approach methodologies, along with 9 other U.S. federal agencies. This was just released last month. We're still in September, right? Last month for public comment, and the link is also on the slides, which I think will be made available to everybody moving forward. I would say that building off of that is where we're considering with the EPA's confidence framework. The ICFAM document speaks a lot to the importance of context of use and independent review, and then understanding technical characterization, information transparency, biological relevance, and data integrity. And I think we're picking that up and carrying that forward in terms of what we're doing in the EPA competence framework, going a little bit beyond in vitro biological systems to include in silico and in chemical approaches, both human and ecological health decision contexts. The 5 bullets here are also shown in the figure that, the importance of the decision context or fit for purpose, the transparency, reliability, relevance, and then, excuse me, and then, sorry, I apologize. Having well-described uncertainties related to the NAMS. So it's not that they will not still necessarily be some uncertainties, as there are with any scientific model that we use, but making sure that they're well described and understood as best as they can be. My apologies, I went too far. And then finally, the framework development will be informed not only by the NES report that I mentioned, the ICFAM work, but also some OECD parallel activities, including updates to guidance document 34, and then the new OECD QSAR framework, which is in development. And then also some of our work internationally through the APCRA case studies, or accelerating the pace of chemical risk assessment. And I had mentioned ICFAM activities as well as others. So this goes back again to the importance of communicating and collaborating and engagement with various stakeholders across the board. So again, going to effective engagement and communication with stakeholders being essential. I mentioned earlier about the upcoming training on HTTK, and this is just a little bit more detail on the work encouraging NAMS use and adoption. And again, I think a lot of it is not only just making people aware, but also educating them on what we're doing and why we're doing it. So, and again, this is obviously specific to some of the EPA work, but it goes beyond that as well in terms of engaging across our stakeholders and working collaboratively across the federal government with other partners. We have the EPA NAMS training. I won't go into all the bullets here, just looking at time, but also the EPA NAMS conferences. We have public conferences every 2 years. The most recent was held last October. So we expect the next one to be anticipated for fall of 2024. And again, there's some links on these slides for follow-up to get, not only to sign up for the training, but also to look at the recordings of past efforts. So what else are we doing when we talk about sort of developing to fill data gaps? Some of that is not just developing new methods, but refining existing methods to address key information and capability gaps. This slide I'll go over sort of at a high level, but there's a lot of work being done by a lot of great people, not just at the EPA, but beyond. And so my hat's off to all of them, because it's a fascinating space to be in. It's just very innovative and able to sort of take what we're doing now and sort of move the ball forward is just one of the best things about my job, I think. So EPA is evaluating or developing a variety of new testing tools to address information and data gaps. It includes high throughput screening and high content methods, so like genomics or proteomics, as well as some conventional methods that aim to improve understanding of toxic effects, looking particularly at TK or TDE knowledge for substances. This slide here shows sort of, it's not really a flow chart, but looking at, puts into context the importance of sort of that fit for purpose piece, the decision context being important, to make sure you know what you're asking before you get started, so that you are going to get the answers that you're looking for, not necessarily predetermined, but that you're able to address the question. So looking at on the left, oh, sorry, I'm on the wrong screen with my arrow, the broad coverage technologies and models. As you go through, you can do integrated combination of technologies and models, much like the IATAs from OECD. And then is it going to be non-selective or selective? And so that would change based on, what are you really asking? If it's non-selective, you could do more bioactivity based and it's more of a predictive result. But if you want to be more selective and you want to be more targeted, then it could be more of a pathway based and be more predictive. And so some of the examples shown here are the different types of methods and models that we've been building and working on and refining that could help address these two, different types of questions, really. So being very aware of what you're asking from the beginning is important. We're also developing bridges from the old to the new methods. So this is a new effort of focus in building on what is strong and credible sort of existing science in the field, in this case of genome sequencing technology and research. We've developed a potential new assessment product based on transcriptomics. So there's a lot of data and support for the transcriptomics science that's available. The study paradigm follows the principles of 3 Rs to refine chronic toxicity testing by reducing the number of animals needed to assess a reference value. It could enable development and release of a chemical assessment of data poor chemicals in as little as 9 months. And every time I say that, we're very cautious about putting numbers like that out there, but that's 9 months from chemical procurement to assessment release. So that gives us that window. Sometimes procuring the chemicals, especially if they're data poor chemicals, is not always as easy to do. The potential new assessment has just gone under review by our board of scientific counselors. It's actually still finishing up review, a peer review by the BOSC. Along with a companion document on the value of information case study, which talks a lot about, what is the value of having data sooner, even if it has more uncertainty. And that's just really, that is a very high level nutshell of that document. But these 3 documents shown at the bottom of the screen are what are undergoing BOSC review. And one is the methodology, and then one is the case studies, and then the value of information document. I highly recommend. They're publicly available on our website in the draft form, and then we look forward to these being finalized and being able to apply them to some of our regulatory decisions. This is a space where we have a lot of data poor chemicals, and trying to find ways, credible ways that we can have defensible science to address that and have some sort of reference value to make decisions is the direction that I think we need to be going in more and more. The next slide here is, evolving the validation and reporting processes to be performance-based and NAM compatible. So we talk about the need to better, build scientific confidence by, if we want to use the word validation, or how we evaluate performance, but we need a way to report that and to be consistent in how we're doing these processes for performance-based review. And so that's what these documents have here. The EPA NAM Work Plan 2024 deliverable will develop the scientific confidence framework I mentioned. NICEDM and EPA are U.S. leads for a new effort through OECD to revise their guidance document 34, which is on validating new test methods. I have a very sensitive mouse today. And then OECD project to develop the omics reporting framework. So really this gets back to the importance of communicating engagement and being consistent in terms of sharing information and how we're able to share that information. We're also working to develop NAMs to impact the needs of the regulatory community. And this is an example of recent EPA efforts to develop NAMs in service of the endocrine disruptor screening program. This resulted in a new 2022 proposal that's out for public, has recently been out for public comment. I think the public comment period has ended. It was released in December, so I think the public comment period has ended. But this is looking at, I think many of you might be familiar with the estrogen receptor pathway model, which is based on 18 in vitro assays from ToxCast and Tox21 battery, and then also the androgen receptor pathway model, which is based on 11 in vitro assays. And so the proposal is that these, for the ER pathway and then for the AR pathway, these could be alternatives to performing some of the EDSP tier 1 screening assays. And then this slide is just another set of, I'm just looking at the time, so I can't see Cecilia's face or if any hands are up, but examples of recent accomplishments for in silico tools. So I won't get into this. Hopefully you're familiar with some, if not all of these. These are some publicly available tools where we try to bring to folks the work that we're doing in a way that's useful and helpful. We generally have training available for these, as well as available on the website for recordings, in order to have a better understanding of the work available and how these tools could work. And then my final slide is just sort of circling back, what are the biggest challenges in the development and use of NAMs from a regulatory perspective? I put some here and I touched on some of these today. Performance evaluation of methods and tools, building confidence and demonstrating application for regulatory decisions. Tools interoperability, I didn't really touch on today just because of time. And then data management and sharing is sort of the overarching need that we see in this space, given that we're able to develop so much more data. And how do we share that in a way that it's usable for a broader group of stakeholders? So I'm going to leave this slide up, but see if there's any questions, and I will hand it back to Cecilia.
Thank you so much, Maureen. We have one question in the Q&A, just asking when will the registration for the HTTK training be up? So I'll let you type the answer.
Okay, I can do that. And I want to stop sharing so I can see people. There we go. Thank you. Yeah, no, I'm happy to do that. So the question is in the Q&A. I'll make sure I get the right dates and put that in there.
Thank you very much.
All right. Thank you very much.
Okay. So if there's no other question, I'm going to move on to our second speaker. Our second speaker is Dr. Steve Musser, who is the Deputy Center Director for Scientific Operations at the U.S. FDA Center for Food Safety and Applied Nutrition. He manages all scientific operations related to cosmetic safety, color certification, pre-market review of food additives, food contact notifications, and food derived from bioengineered plants. He also directs research in precedent-setting areas of food and cosmetic safety. So today his talk will be on the importance of identifying regulatory gaps prior to tool development. That's a very important topic. Dr. Musser, thank you for being here today.
Thank you, Cecilia, and thank you for the opportunity to speak this morning. I really appreciate this, mainly because it's an area that we've seen really exploding. Lots of questions, a lot of interest, a lot of money being spent in development in these in NAMs and alternative methods, and for a lot of good reasons. This has been something that's been on our mind as well as EPA, and I would really like to thank Maureen for giving such a great background. Many of the things that she discussed from a context of EPA is very similar to things we're thinking about at FDA and trying to integrate. We have some differences, but in many respects, the approaches are very similar. And so really want to talk a little bit more in detail about some FDA specific things and the way that we're thinking about it today. So when we think about FDA, people often think about the medical products, the drug side, devices, biologics. I'm on the food side, the Center for Food Safety and Applied Nutrition, which will soon be known as the Human Foods Program. We're undergoing a reorganization that's been very publicly announced by our commissioner. So we regulate a lot of products, foods, and also cosmetics. And I wanted to give you a sort of an idea of what that regulatory space is, because I think it's particularly relevant to gut health and issues related to the gut. The center has seen a lot of not only contaminants that might be environmental contaminants, but new foods, micronutrients, dietary supplements, cosmetics, while not generally eaten. This tends to be often a problem with children where they may be eating cosmetics. You don't think of that very often, but it does happen, and something that we do want to think about in terms of how all of these different areas that we regulate fit into a NAM space and our regulatory space in general. We have been getting lots of questions about the use of NAMs and how we integrate that into our regulatory space. And I think from our perspective, it's still a developing area. All of the centers are in various stages of developing guidance or publishing guidance on the use of NAMs and integrating NAMs into the regulatory workflow. Because the centers have such very different approaches and different ways of regulating products, for example, the difference between food, drugs, and tobacco, very significant endpoints, very significant types of measurements that are being made. And while there may be a lot of similar areas that would be covered by guidance, the specific areas that particular center covers is going to be a little different. So we'll be getting guidance out from these centers as it's developed and approved. So at the center, when we at food in the food space, when we think about where are the gaps and how are we developing these approaches, I thought I might bring up some recent issues that we've developed or we've noticed, and that might be of interest to this group as you think about developing NAMs and how those NAMs could be useful in a regulatory context, or might be useful in submissions that come into the agency. One of the new things we're seeing more and more of is novel foods. And these novel foods are often plant-based proteins that are used to supplement or completely replace animal proteins. So we're going to see often very high levels of proteins that people haven't been exposed to normally. So for example, let's say it's a pea protein or a potato protein. Now, most cases, people aren't going to have very high exposure to pea or potato protein, but in some of these replacement foods, they're the primary protein that's present in that particular food, and so you might see allergies that would not be normally present, but are now present because of the very high levels of these proteins. They're not new, they're not novel, but the proteins themselves are present in much higher abundance. So there are digestion issues, there are allergenicity issues associated with them. And then we have completely new food sources that are just being developed. There's a lot of venture capital, a lot of new companies, small companies often in these spaces where a new fungus might be developed that's grown in a special way. They've come from very different sources. They wouldn't be fungus that would be typically traditionally used in a food source or available in a food source. And so there's issues of unknown toxins that may be produced, secondary and tertiary toxins produced in the fungal process that would need to be looked for. There's insect protein is becoming, and while it's been available in many parts of the world for centuries, insects are now becoming something that people are really thinking about supplementing proteins for humans on a larger scale. Not a lot of data out there on allergenicity impacts of these proteins and insects on other parts of the gut or the body, and so we just don't know. And so lots of gaps in our understanding about some of these new food sources. Then we have genetic engineering and genetically modified crops. You're all aware that this is going on. And so are there unintended consequences that may happen if we look at a particular oil or a particular protein that's modified? The product that is trying to be achieved is actually, the phenotype has been developed and it's there, and the company has the right thing. Is it safe, or is it having some unintended consequence somewhere in the body or in the some other part of the food chain? And then lastly, and this list could go on and on and on, but these are just some of the larger areas that we've been looking at. Dietary supplements continue to be a very large and growing portion of our regulatory portfolio, particularly live microbials. There's a lot of research now being published on the impact of these microbials and the microbial gut interaction in community, where there's all sorts of secondary or primary effects caused by everything from Alzheimer's to celiac disease, numerous disease. And I'll also let you know that from food's perspective, we're really only looking at safety. We're not considering efficacy. So when we look at the NAMs and the way NAMs are used, we're not looking at the safety or the efficacy of these microbials, only the safety. Are they safe in the gut? And then infants, particularly preemies, we've had a lot of very unfortunate incidences where live microbials have been given to these premature infants without fully developed guts that have had quite tragic responses. So obviously, we would like to have models that could prevent that, or that we could evaluate the safety of these types of products and help prevent any of those circumstances from happening again. So when we look at NAMs, and if you look at a traditional approach that the agency and the center has looked at for decades, probably since inception, you have your chemical hazard, you have a gold standard animal tox type of approach, and then an estimate of human risk and safety. And what we're jumping to now is we're skipping this animal, or trying to skip this animal toxicity data step. And there's some really good reasons for doing that, not to mention, especially in the case of gut, there are ethical issues with using humans. Animals don't often correctly mimic the human gut. There's a lot of really good reasons for not using animals or replacing animals with other types of testing. So you have in vitro and in silico methods for estimating the safety and toxicity of a new food or a new chemical that's introduced into the food supply. So how do we skip that animal toxicity data piece and get to estimating human risk and safety? All right, so now we're into the NAMs and the NAMs approaches. So FDA has really had a longstanding commitment to development of these new technologies. Every center in the agency is developing and evaluating these tests, either in-house or through contracts or collaborations. In our case, we do a lot of collaboration with EPA, NIH, other worldwide agencies and groups doing work in NAMs. I think Maureen mentioned ILMRAC. We're a member of ILMRAC. We regularly participate in those types of approaches. We've done a lot of, or tried to do a lot of work in this for over a decade. We do believe that these approaches will not only provide safety or a measure of safety that's not available anywhere else, but will help prevent products with increased toxicological risk from reaching the market. And that's the safety piece that we're really most concerned about. We don't want any of these new products or old products, even chemicals, to have effects that are unsafe to the population. Also, I did want to mention something about outreach and workshops and education. We are just beginning, at least on the food side, and I'll announce that we are going to have a little workshop on November 14th and 15th in College Park, Maryland, on developmental neurotox, and perhaps not of great interest to the people here doing just gut type of work, but there will be a workshop in person and hybrid type of event in College Park, Maryland in November. So we do have these NAMs investments, and we've talked about, both Maureen and I, replacing, reducing, and refining animal testing. And I would say that our primary interest is better modeling of human toxicity. We have really seen where the gold standard animal test is very, very effective and has been for decades. But there's a lot of holes. There's a lot of gaps. There's a lot of places where we could improve our knowledge, even so far as designing better animal experiments using these approaches. And one of our real issues is, how do we get better human information? How do we get safety information that's particularly valuable to evaluating the safety of these new products and chemicals, proteins, other new foods in the context of human toxicity? So there's also the promise of lower overall costs. And I will tell you, as someone that's been paying for some of these new evaluations, we haven't seen the lower overall costs yet. Now, hopefully that happens, but this is an area that is a gap. Many of these methods are quite expensive, and we are looking for ways of reducing the overall cost, at least from our perspective, although we recognize that in its infancy, we will have cases where these these methods are going to be more expensive. All right, so we have lots and lots of alternative methods. There are new journals coming out that just deal with alternative methods. There are in silico, alternative organisms, in vitro models. There are just I would say an exploding area of research with pretty much anyone that has an interest in this starting to develop methods, which is okay. But how do you pick the ones that are useful from a regulatory perspective? It's certainly not enough to just have it published in a peer-reviewed publication. A good start, but we would like to see a little more. We'd like to know a little bit more about those methods as we gain confidence. And I really like Maureen's definition and the description of where these methods are headed. I really do believe at this point it's about gaining confidence in these methods. There's a lot of them out there. Which ones do we have confidence in? Which ones do we feel as a scientific community and as a regulatory agency, which ones really provide the confidence we need to make decisions? We're talking about people's health. We're talking about products that have been in development for decades. How do we ensure that those products are safe and that public health is safe? So to that end, here's our challenge. The major one being relevance. Yes, you've measured something in a system, but what are the endpoints that you're measuring? And are they really predictive of in vivo effects in humans? Or can they only be described in a particular confined environment with that particular test? And do those results account for relevant PBPK and exposure estimates? One of the most significant issues that we've been grappling with is we see, let's say, for example, a receptor binding assay or a very specific assay, but it's in the absence of any exposure data that's on that particular compound or measurement. And that's difficult because we have to figure out, well, you've seen an effect in a system, an isolated system. Is it really relevant? Is there enough there? How much is there? At what level do we really see some kind of measurable risk that might occur from that in vitro test to a human type of exposure assessment? And so making that leap and understanding that gap has been a very significant jump for us, and we could really use some help in modeling and how we take those efforts forward. And then, is it translatable to human and at-risk groups? So when we tend to think about animals, you're thinking about, well, I have this one set type of animal that I use for this type of experiment, and often, as I've noted earlier, they're not really translatable to humans. But within humans, there's a whole span of people that have lots of different types of risks, and one of them could be small infants, and the other significant at-risk groups are older adults. And those groups, how do we evaluate safety in young, very young children, often premature births, and at-risk older generations, 65 and older? Those tend to be different than your standard type of experiment that might be done, and something that we really need to look at carefully. I mentioned Alzheimer's, gut health on Alzheimer's, a number of publications on that. So you imagine the target audience here that these types of dietary supplements would be sold to. We'd really like to make sure that they're safe. Okay, moving forward, just some specific things that have come up as we've looked at these and seen submissions that where people are using NAMS types of approaches is the assay mature enough? Do you really understand the platform? Is it stable? Are the cell lines reproducible? We've had substitutions of reagents which cause very different outcomes. Suppliers aren't qualified, so you've got you're using different suppliers. You might have one person using one supplier and another person using another supplier. And while it might be the same type of reagent, you get different results. So we really need to understand that. Is it reproducible, both in a single lab and outside of another, in this one lab and in another lab? How much confidence do we have that that type of data can be found in more than one type of laboratory and in the same laboratory? Do you have a good understanding of positives and negatives? What works in the assay? What doesn't work in the assay? People often focus on, well, we have these one set of compounds that work really well, but what doesn't work? Does it stick to the plastic? Is it soluble? There are all sorts of other considerations when looking at the qualifying one of these types of tests. How does it compare to in vivo data? Have you taken compounds where there is good quality in vivo data available that we can compare and get some measure of confidence based on that type of data point? And then lastly, define the limitations of the assay. What are the sensitivity and specificity of the assay? And to some extent, this gets into the issue of exposure. What are we talking about when you see a response? How much do you need? And how is that translated into a human dose or a human exposure assessment? And that kind of information, it's not just enough to see a response in an assay. We need to be able to translate that into what effective dose a human may be getting. Okay, now this slide is a bit busy, but what I'm trying to explain here is that we're essentially with NAMs, we're taking apart an animal into individual components, and then at some point trying to stitch all of those components back together and get an idea of the whole animal. So we've got artificial intelligence, data integration, in vivo models. How do we get that all back in a meaningful way so that as a community, we agree that that's how it would be done, and we would see that kind of representative response based on those types of inputs. This is an area where there is a tremendous gap right now. People are pouring money into AI and rightly so. Also, data modeling and in silico approaches to moving all of this information into a system that can be understood and made relevant to the organism, the whole organism. Really important that we figure this out while it's still early and make sure that as a community, we really understand how this type of data integration modeling of information from NAMs is really going to work. So in conclusion, there is a significant need for modeling and new approaches to assessing human toxicity. We all recognize that. That's why we're here today. And you're listening to these types of workshops. Really, actually very interested in the number of the follow-ups. I think this will be a very good workshop and something I'm really interested in listening to, and I know a number of people at FDA will be tuning into as well. I do think we need, as a community, a greater emphasis on qualification. I would say validation, but that's kind of specific to certain FDA terms and needs. And at least at this point, if we could just get to how would we qualify these? How do we move them from beyond a single laboratory that can do this type of work to collaboration and comparison across laboratories, so that more than one person or one laboratory has real confidence in what the outputs of these methods and approaches are going to yield. And we don't get into arguments where one laboratory and another laboratory are arguing that their test is better. That's from a collaborative and comparison approach really makes regulatory decision-making difficult. And lastly, another plug for data integration. We should really be working on this now. I know a number of people are, and I do think that how we integrate all of this information and use it is going to be key to moving ahead. So that's it. Thank you very much for inviting me and for listening to me this morning. And I think I stopped with enough time for questions if there are any. So I'll turn it back over to you, Cecilia.
Thank you so much, Dr. Musser. We do have two questions for you. The first question is about the DNT workshop in November. Could you provide more details?
Yes, we'll be sending out an announcement and posting it on our website soon. We just finalized the agenda. It will be, let me look at the announcement. It will be November 14th and 15th. And you can contact me, or you can contact Omari Bandelli, who is organizing the meeting. Its last name is Bandele. I'll put it in the response here. And you can contact him directly. Okay.
Thank you. Thank you. I'll let you finish typing before I ask you the second question.
Okay. Okay.
Okay, so the second question is related to the cost. So it's about the fixed and variable cost. The question is, how much of the current high cost of NAMs is a result of equipment and other one-time purchase to build an infrastructure for NAM work? Will the price benefits start becoming more apparent once those overhead costs are accounted for?
Wow, really good point. Someone that obviously, has thought about this a lot. I think the answer is yes. And one of the problems is scale and reproducibility. So currently, there are very few of these methods that are done in more than one laboratory. And so I believe that once you end up with a standardized approach and let's say you have a standardized approach for some kind of developmental neurotox approach. I know OECD just approved a DNT assay. So once that starts to be integrated and more labs pick it up, and there should be a reduction in cost based on more laboratories doing the same thing. While everybody's doing something different, it's just going to be expensive.
Thank you. And then one more question. NAM means uncertainty to the regulators and costs to the industries. So what are the benefits?
I think that, at least the primary benefit as we see it is that you get a much, if it all works as, advertised, and I certainly believe that it will, the real value is you get a much better picture of what's happening to humans. I think we have all wondered, yes, we have these really good gold standard animal methods for measuring all sorts of tox endpoints, but and we know in many drug development cases that they haven't been the best way of assessing human toxicity. I just feel, and many of us feel, that it will give us a much better understanding of the human condition, especially as I said, humans they span a lifespan of risk, and you have infants, you have adults, you have different ethnicities. And I think NAMs, when fully explored, will give us a much deeper and richer understanding of human toxicity than we've had so far.
And I don't know if Maureen, you would like to add more?
No, I agree. I think Steve answered that very well. So nothing really to add there. Thanks.
Thank you. Okay, so I think that's it. Let me see. Oh, there's one more question. Okay, qualification testing is costly. Mostly about the cost today. What types of mechanisms can help assay developers conduct these studies?
Yes, kind of the next step. I think as I was just in Europe, I was talking to folks at EFSA about this. I think as regulatory agencies, we need to take a leadership role in trying to figure out how to set up mechanisms, either through granting process or working with NIH to take that next step and help developers, especially small developers and small businesses approach this qualification. It is expensive, but it's necessary. Unless we get some more confidence in these tests, I think we're going to be having difficulty figuring out how to accept them. I don't know, Maureen, if you have any programs at EPA where you're qualifying things or working on that type of approach.
No, I mean, I think the terminology is a little bit different. We're not really doing sort of qualifying testing at the EPA. So I think, yeah, it's just slightly a different lens on that. We do have some activities that we're doing where we're looking at, evaluating the performance of our NAMs, but I think it's a slightly different question. That seems a little bit more specific to the work that you're doing, at least in the ORD. I don't know, Cecilia, if in OPP or OCSPP, there's something along those lines. Okay.
Okay. So it looks like that's all the questions we have today. I would really like to thank Steve and Maureen again for sharing your knowledge and insights on this topic. And before we part ways, I would like to remind everyone to mark your calendars for the other two webinars and then also the in-person workshop. Thank you all for your engagement today, and we are looking forward to welcoming you back for our next webinar. So wishing you all the wonderful week ahead. Thanks again.